Good morning, and thank you for this invitation to talk today. So I'm a hepatologist, a liver specialist, and my interest in the Raine is actually related to fatty liver disease, and we've looked at that in the 17-year and the 27-year cohort, and we're really looking at associations and causes of fatty liver disease, and this really led us to look at the microbiome as well. And so the analysis of the microbiome within the Raine cohort is really in its infancy, and so I don't really have any data to share about that, but we'll be discussing why we're looking at the microbiome and the paradigm of how to use it to look at other disease phenotypes or health outcomes within the Raine study. So just what are we talking about when we talk about the microbiome? And so they're not actually one and the same, microbiota and microbiome. So microbiota refers to the actual organisms themselves, whereas the microbiome refers to the DNA, the genome, the building blocks of the microorganisms. And this is how we actually identify and characterize the microbiome, by looking at its DNA. And it's very varied, and so within the human, you have different repositories of bacteria and other organisms, and they vary depending on what site of the body they are. Now, as has been alluded before and this morning, the microbiome is incredibly large and diverse. There's some fun facts about it. There's actually 10 times as many microbial cells in your body than human cells. And the genome associated with the microbiome outnumbers your personal genome many-fold. It's very complicated. There's greater than 10,000 different species of microbiota living in and on you at the moment. And there's a lot of variation in this species of microbiome between individuals. So I share 99% homology in my genetic DNA with Romola, but my microbiome is 80 to 90% different from hers. And so this variability and complexity in the microbiome is challenging when it comes to analysing the microbiome. So what does it do? So I'm going to focus now on the gut microbiome because that's what we've got samples on. So the gut microbiome essentially is our interface with anything that we put in our mouth. And so this includes obviously diet, but also drugs. It interacts with our immune system, and so to avoid us being overridden with bacterial sepsis or viruses, there's a delicate dance between our immune system and the thousands and millions of various bacteria within our gut. And being bacteria, we'll focus on bacteria. Being bacteria, they produce products, and these products can be absorbed into the blood system and lymphatics and have effects on distant organisms. And so what are the impacts of these functional changes of the microbiome? Well, it changes what nutrients, micro and macronutrients, that our body sees. And so there's actually different harvest or different efficiencies of extracting calories from what you eat based on your microbiome. It impacts on our bile acid metabolism, so bile is made by the liver. One of its jobs is to help digest food, but bacteria also metabolises bile or bile acids to produce what we call secondary bile acids, which are reabsorbed into our circulation and have distant signalling effects on things like muscle, liver, heart, brain. And so you can see how the gut microbiome might be distally affecting other parts in the body. The microbiome also stimulates hormone release from the intestine, and again, these hormones have quite wide-ranging effects on different systems within the body. They're involved in immune development and also nerve signalling. For example, some of the molecules involved in depression and anxiety, serotonin, etc., all come from metabolism related to the microbiome. And so there's been a whole host of different organ systems and diseases which have been implicated with the microbiome, and I've put just some on this slide as an example. And so the evidence probably is most advanced in the cardiometabolic world in terms of obesity and type 2 diabetes, but you can see that there's been diseases within the lung, the brain, heart, the skin, and there's many more beyond this slide as well. So is it really causal? Is the gut microbiome really have a significant effect on our health? And really there's some seminal studies that came out in the mid-2000s which really changed our way of thinking of how significant the microbiome was. And this was associated with our ability to produce germ-free animals, i.e. mice that didn't have any bacteria or within them. And so this particular study, one of the early studies, took lean mice and compared it with genetically obese mice. And mice are coprophagic, so they love to eat each other's poo. And so it was, through this study, they were able to transmit their microbiome from the lean mouse to a whole group of other mouse. And they found that if you fed the microbiome to other mouse, that you could actually transfer obesity. And this was actually independent of the amount of food that they ate. So this was proof of principle. But we're obviously humans rather than mice. So how can we use the gut microbiome in the Raine cohort? So we can think about using it as a diagnostic tool. We can look at pathogenic factors for disease. And by doing that, identify treatment targets to try to change outcomes, improve health outcomes. And also we can hopefully use it to maximise treatment effects. So you may remember, some of you at 27, you got a kit at the assessment and asked to go home and deposit something into that kit, put it in the post bag, and then that came to us. And what we've done with it so far is we've extracted the bacterial DNA from that, and it's just about to undergo bacterial sequencing. So what questions are we asking in the Raine cohort? Well, firstly, we just want to characterise it. What is the composition and diversity of the gut microbiome within the study? And most importantly, what are the determinants of the gut microbiome? We've got a whole wealth of longitudinal data, and what are the factors which are important in shaping your gut microbiome? The next question is really to associate that microbiome with different outcomes. And my particular research is fatty liver disease. And so what we're looking at is how diet may influence fatty liver disease through the microbiome. And so this paradigm can be used for multiple diseases. What is an environmental triggers or exposome which causes a certain disease or health phenotype, and how does the gut microbiome modulate that? And so I actually have too many slides, so I'm going to skip through these. But the rationale to do this really came from our earlier work where we had diet. We've looked at diet at age 14 and found that it was associated with an increased risk of fatty liver disease at age 17. But there was variability within this relationship. So people who are 14-year-olds which were consuming a Western diet or a high fructose diet, although there was an increased risk, not all of them actually developed fatty liver disease. And so what we're hypothesising is that it may be the gut microbiome which is mediating or modulating this risk. So hopefully we'll be able to then develop an idea about precision or personalised nutrition, where individual characteristics, including the gut microbiome, may be able to determine your response to diet. I might just skip over this study for the interest of time. This kind of work cannot happen in isolation, and there is a large group of collaborators, both national and international, behind this effort, and I'd like to obviously acknowledge them, but also obviously thank you for the Raine participants for donating their poo to me. The study staff, particularly Alex and Di, for doing such a good job at convincing the participants to donate your poo to me, and also my staff for processing it. So thank you very much.